Coming up on the FSR Sarc Fighter podcast, promising sarcoidosis research for your whole body that starts on your skin.
My lab is really good at modern scientific techniques, including single-cell technologies. And so these technologies allowed us to look at what the individual cells within these thousands of cells, what they were and what they were doing within the skin sample.
Dr. Thomad Leung is diving deep into the mechanisms that cause sarcoidosis.
And then the second finding from this result that we find that's really exciting is that we discovered that these ILC1 cells are recruited to sarcoidosis granulomas through a specific homing signal called CXCR4, which is a receptor that sits on the cell, and it actually just, the cell basically follows that, uses that receptor to follow what signals and how to move to different organs. And we think that potentially by blocking this receptor, we may have a way to disrupt granuloma formation and accelerate granuloma resolution over time as well.
More of Dr. Leung's fascinating research coming up.
This is the Sarc Fighter podcast, living with sarcoidosis and other rare diseases. Here's your host, John Carlin.
Hello, this is episode 151 of the FSR Sarc Fighter podcast. I'm your host, John Carlin. The date of this recording, November 16th, 2025. And I had another take that sarcoidosis moment, like take that sarcoidosis, last weekend when I got on my bicycle and I rode my birthday in miles. It's the 5th year that I have done this, starting when I was 60, so you can do the math. It's a little daunting because every year you add a mile to the distance, and of course you're another year older. So your ability decreases just a little bit because of age, and the distance increases just so incrementally, but eventually those two lines are going to cross, and you're not going to be able to do it. So you do this, it's just something that I'm determined to do. You do it on the date of your birthday or as close as you can. And my birthday did not land on a weekend this year. And then I had to navigate a lot of work commitments, some family commitments. The weather was particularly bad, but last Sunday, all the planets aligned. I had a clear shot at it, and 4 hours and 51 minutes later, it was in the books. So there you go. And you can look me up on Strava if you know what that is. If you know, you know. And you can see all of the data if you are really interested in that. But it's kind of a challenging ride. There's a lot of climbing, a lot of hills. There's one kind of long mountain that's gradual, but it goes on for several miles. And so, I'm pretty proud of this. But as I've said before, when I complete a goal that has a certain degree of physicality related to it, I say, take that, sarcoidosis, and this was one of those moments. And, so like you, I can't mourn the loss of my old life when something like this probably would have been a whole lot easier. I can just embrace my current one and have gratitude for what I can still do and what I can still enjoy. And speaking of that, after the ride, we had a small birthday party for me. My wife threw it with family, and 4 of my grandkids were there, and they helped me blow out the candles. And, that kind of stuff, that's among those life moments that you really have to learn to cherish and respect and see them for what they are. And I will remember both of those things from that day forever. Having completed the bike ride and just spending that quality time with the grandkids. And then as they were leaving the house, I was sitting in a chair in our living room, and somebody said, "Give Papa John a hug before you go." And all 4 of them just jumped on top of me and gave me a big hug. And that was really, really special. So, all right, let's turn to the matter at hand, shall we? I am amazed at how much research is being done on sarcoidosis, and all the progress that's being made in our battle to understand the disease, and how many highly qualified researchers are now in our space, and they're all chasing these answers. And I'm going to say that in two contexts related to conversations I've had in the past week. First of all, my guest today, who we'll hear from in just a few minutes, Dr. Thomad Leung, who is using the skin to detect and study sarcoidosis anywhere in the body. You're going to hear him explain what he's doing and how he's helping our understanding of the disease, getting right down to the molecular level. So don't dismiss this as, oh, well, I have sarcoidosis in my lungs, or, I have it in my heart, or, my spleen, whatever it is. What he is doing could solve the puzzle no matter where the sarcoidosis is. So now, as we basically know, the autoimmune disease, any autoimmune disease, is when the body attacks some portion of itself that doesn't need attacking, doesn't need help, doesn't need all of these resources rushed to the joint or to the lungs or to the skin or whatever it is. In our case, of course, it's these granulomas that form in the lungs, the heart, the spinal cord, you name it. And when they do that, they actually, instead of helping, do damage to whatever organ they attach themselves to. And we don't know exactly why this happens. Well, Dr. Leung is going to explain how they're getting very close to discovering whatever it is that sends out what he calls a homing signal to all those T cells and macrophages that then come running toward the signal and form these things, these granulomas that hurt us wherever they form. So then if you carry that forward and we can figure out what's sending the signal, then we can find a way, hopefully, to block the signal with some kind of new medication or therapy, or maybe a medication that already exists. And then we're on our way to finding perhaps a definitive cure for sarcoidosis. Now that's me, John Carlin, supposing. The doctor's not saying that. FSR is not saying that. We're not saying we're on the precipice of a cure. I'm explaining to you the value of this research and why it's so important. So that is a long, long way off, but Dr. Leung is making progress, and you're going to hear him describe that in a way that's very user-friendly. I don't think, even though, he can get downright granular when he starts talking about medical stuff, but he did a really good job in explaining this in a way that I could understand it, which I take to mean that you can also understand it. Now, I also, in this same vein, I want to talk about how FSR is funding other research. In my role as a new board member, I and my fellow board members had to vote on who would be receiving funding for their research in the latest round of FSR grants. So, researchers say, okay, they've got an idea or they're making progress on a concept, and they submit funding requests with all the justifications on why their work should be picked so they can continue to look at whatever it is that they're looking at. And these submissions are reviewed and scored by staff and a committee of medical experts long before the board ever votes. And they're ranked with justifications and so forth. And then we as a board have to figure out who makes the final cut. Right? This was my first time participating in the process, and I have to tell you that for me, this was hard. It was hard because there are so many wonderful submissions, and you don't have enough money to fund them all. So, in my opinion, we have a blue ribbon board of directors. There's a lot of medical expertise on the board. There's a lot of people who have been in the sarcoidosis space a lot longer than I have. And so what I did was I sat back and I did a lot of listening. I asked just a few questions, but, I was still going to have to vote at the end of the day. So, think of this as like, let's say there's a new congressman, right? And they got angry and they decided, you know what, I want to I want to go to Congress and I want to fight for my district and I want to fight for the things I believe in. And then they get there and everybody else has been there for God knows how long, and you're the rookie, and you still have to vote. You still have to make a decision. So that's kind of where I was, right? But when I say that this podcast is all about answers and hope when it comes to sarcoidosis, this is what I'm talking about. Now, I can't reveal anything yet about what was approved or not approved. That news release will be coming out from the good folks at FSR. It'll be distributed through proper channels, and then I can talk about it. Then we can look at what these other researchers are doing and give you more reason for hope. And then perhaps down the road, maybe one or two of the researchers will agree to appear here on the podcast. That has happened in the past. So the reality is that there's a lot of quality research happening out there, that FSR has been very successful in fundraising for the fight, and then a portion of that money is funneled back into research. Some goes into helping patients, some goes into maintaining the engine that keeps this thing driving. It all goes to very important aspects of the lives of people who have sarcoidosis. But a certain amount of that money is funneled back into the research that shows great promise in unraveling the mystery of sarcoidosis. And then, so toward that end, Dr. Leung will be joining me next here on the FSR Sarc Fighter podcast.
I feel like a zombie, just feeding and stumbling.
Hi, I hope you're enjoying the Sarc Fighter podcast. You may be wondering, what can I do to help? How can I be a part of the sarcoidosis solution? It's simple. Make a donation to KISS, Kick In to Stop Sarcoidosis. 100% of the money goes to the Foundation for Sarcoidosis Research. Look for a link in the show notes of the Sarc Fighter podcast. Welcome back to the FSR Sarc Fighter podcast. Joining me today is Dr. Thomad Leung, who is an associate professor of dermatology at the Perelman School of Medicine, and he's also an attending physician at the the Crescents VA Medical Center? Did I say that right?
That's correct. In Philadelphia.
In Philadelphia. Okay, great. And Dr. Leung, you are using dermatology as a proving ground for precision medicine all across immunology, including sarcoidosis. What does that mean?
So yes, thank you so much for the opportunity to be here today and to share some of our stories. So I am a dermatologist scientist. I run a research group where we study how skin and the immune system responds to injury and inflammation. So why the body heals sometimes perfectly and other times why that same process can go off track and cause disease. And one of the advantages of being a dermatologist is that the skin is much more accessible to the outside world than other organs like the lung, heart, brain, etc. And so we are trying to take advantage of this by saying, hey, if we study how disease processes work in actual disease tissue, we might understand the mechanisms driving this, and these mechanisms might be applicable to these same diseases that happen in other organs like the heart or lung. It's more difficult to treat, to study organs. And so that's why we think that skin might be a great way to study diseases like sarcoidosis.
Well, and of course there's a fair number of people who have some sort of a rash at some point along the way with sarcoidosis. Obviously, it's easier to biopsy a rash than it is lung tissue, for instance. But is there anything in particular about the skin that also, other than ease of access, that says, you're onto something here in terms of total immunology?
So I think the best I think the best way to illustrate this is with concrete examples. And so
Sure. Yeah.
30% of sarcoidosis patients have skin findings. It's actually the second most affected organ in sarcoidosis, lung being the first at 70% of patients having some sort of lung involvement. And so by biopsying directly diseased tissue and comparing it to non-diseased tissue within the same person is the perfect control for almost all experiments. So skin helps us set up a really good, well-controlled experiment to figure out how things are going. And so what I did was I collaborated with a really good friend and colleague, Misha Rosenbach, who is a skin sarcoidosis expert at the University of Pennsylvania, and runs one of the largest skin sarcoidosis centers in the country. And so we decided to collect skin from 28 different patients with either skin sarcoidosis or other skin granuloma containing diseases as a control population, as a comparator. And we use these samples. My lab is really good at modern scientific techniques, including single cell technologies. And so these technologies allowed us to look at what the individual cells within these thousands of cells, what they were and what they were doing within the skin sample. And what we found was really exciting. The first question we asked was, how does a skin sarcoidosis granuloma different from any other skin granuloma that we might be seeing in these diseases? And what we found in sarcoidosis granulomas was a specific recruitment of a specific immune cell type called type 1 innate lymphoid cells, or ILC1s. And these cells seem to act as organizers of the granulomas by bringing other immune cells into the granuloma, as well as building them up into a more mature state. Excitingly, we also found that, and so the first question would be, great, we believe your work, Thomas, but is this really just a skin-specific phenomenon, or does this really apply to other things as well? And excitingly, we found that no, it applies to everything. Lung granulomas, lung sarcoidosis granulomas, cardiac sarcoidosis granulomas also have increased recruitment of this cell type. And then the second thing that we found really exciting was that even the blood from sarcoidosis patients, specifically compared to other healthy controls, have elevated levels of this immune cell type. And so this finding allowed us to have two different conclusions. One, perhaps these innate lymphoid cells, type ILC1s, might be a biomarker, a marker for sarcoidosis disease that we might be able to track either in tissue or in blood. And what would be even more exciting was perhaps these levels might go up or down depending on how your disease is active or not, right? And so this might be a way for us to track whether medications are effective, if your treatment is appropriate, and things like that. And then the second finding from this result that we find that's really exciting is that we discovered that these ILC1 cells are recruited to sarcoidosis granulomas through a specific homing signal called CXCR4, which is a receptor that sits on the cell, and it actually the cell basically follows that, uses that receptor to follow what signals and how to move to different organs. And we think that potentially by blocking this receptor, we may have a way to disrupt granuloma formation and accelerate granuloma resolution over time as well.
That's amazing. I mean, that is really amazing. So there's a homing signal. So you say homing, for instance, I think of like a homing pigeon, right? Like it always knows where to go. It's got that sense. And so you've found something that sends out this signal that attracts all these things that form granulomas that we don't want to happen.
Right.
And if that's true, and if you can disrupt that, you can undo everything.
That's our hope. That is our hope. And so to go back to your original question of why skin? I think by studying how disease processes really work directly in human skin and human samples, I think that's the biggest differentiator. We're not trying to extrapolate from an artificially constructed experimental system here. We're really saying, look, I'm just going to go zoom right into the most affected tissue, try to figure out what's different here compared to other people with similar skin diseases, and find what's unique, and hopefully we can target that.
Now, so how do you decide what is a similar skin disease? There are other types of granulomas or other things that cause issues like, I don't know, eczema. I don't speak skin disease, but what else do you look at?
That's absolutely right. So we've done it both ways. For this study, we said, look, we know that granulomas are basically clumps of immune cells that can appear in any organ, and sarcoidosis gives us one flavor of these clumps, but there are other diseases that also create these similar clumps. And so diseases like granuloma annulare, necrobiota, like I can name off even more foreign sounding diseases, but basically there are other skin diseases that also contain granulomas that we know that are different than sarcoidosis granulomas. And so we think that's the perfect way to compare. We've also used other common skin diseases as comparators, like atopic dermatitis and psoriasis, things that people are generally more aware of. And I guess that would lead us to our second finding, that our more recent work is ongoing and almost done now. And so I had described how we had collected these skin samples from 28 different patients, right? And
yeah.
did this giant database of trying to figure out what the cell types were within them. And so the first way we decided to interrogate this was asking what's different between a sarcoid granuloma and other granulomas. A second way to interrogate this data set, aligned with what you're just asking, is to ask, well, what's different between all granuloma containing skin diseases compared to all the other skin diseases that anyone can get, like atopic dermatitis, like eczema or psoriasis. And so that's what we've done as a second round of research here. And that work has actually discovered something even more fascinating. And that part of this answer came from something that was unexpected to us, which was the blood clotting system. And so when we asked, tell me the molecular signatures that are unique within all granuloma containing skin compared to non-granuloma containing skin, the top hit was a protein called osteopontin, which is a secreted signaling factor that people have studied in other contexts. But what made this more unique was that we found that the clotting, when your blood clots, there is a enzyme that is critical for that process called thrombin. Thrombin.
Yep.
And so thrombin is an enzyme that will cut larger proteins into smaller fragments. And where this research is going is that we're showing that thrombin cuts this full-length osteopontin into smaller fragments, and these smaller fragments is what keeps driving granuloma formation, as well as maintaining granuloma formation over time. And if you can block either the thrombin itself, or you can block these smaller fragments specifically with antibodies or other things, that we can disrupt granuloma formation, at least in a mouse. I can do it so far.
In a mouse, wow. But you've got this animal model that you're working with, and then you've also done the human stuff. So let me back this out even first of all, I'm fascinated with whatever equipment you use to look at this stuff and how anybody, can get right down to that level. But I want to back out further before I come back to that and ask you, okay, well, do you know what the origin is that starts the whole process? Have you been able, because everybody wants to know what causes sarcoidosis, right? And a lot of people say it starts with the rash. They don't worry about it, and all of a sudden they've got it in their heart, they've got it in their lungs, whatever. But like, what triggers the whole process? Have you been able to figure that out yet?
We haven't, unfortunately.
Yeah.
So I would say that's the next frontier that we want to get into.
Yeah.
So sarcoidosis has been around, like we've been studying sarcoidosis for a century. This is an unanswered question over 100 years already.
Right. Yeah.
for probably the first 75 years, people were really focused on trying to find an infectious cause. This is something that some sort of bug triggered it and caused the immune system to go awry. That hasn't been that fruitful, to be honest. People, not me, but other people have done many years, decades of work and haven't been able to find that. Other people have been speculating that this is a non-infectious trigger, where this is, it could be something, a substance of some sort, or another trigger that's non-infectious that's causing the disease. We haven't been that successful in finding it. We've been successful in linking it to specific things like obviously the World Trade Center dust
Right.
from the collapse has increased the frequency of sarcoidosis in healthcare workers that participated, in the first responders that participated in that tragedy.
Yep.
And so that really suggests that it might be something like an organic particulate matter. And so we have been trying to tackle this by saying, okay, look, we have this immense database of what goes on in skin granulomas. Can we detect any signals that reflect an infectious origin? And even with the highest molecular standards that we have not been able to do that. And so if you had to ask me, I think I favor a non-infectious trigger,
Mhm.
we haven't gotten to the point where I can tell you exactly what that trigger is.
You haven't figured out what it is that starts splitting these cells or these enzymes. I guess you're way below the cellular level, and then okay. I don't want to get out of my depth here. I'll just keep asking the questions and have you explain. How do you look at these minuscule little pieces of biology to track it the way you're doing? What kind of equipment? How could you describe that to us that we would understand?
Okay, so I think this is the age of biology. Like, I think we had an age of physics, we had an age of chemistry, and I think we're now really in the age of biology and making important steps in biomedical research. And so if you asked me this question 20 years ago and said, Thomas, can you please study how granulomas formed and how sarcoidosis works? And even if we said, hey, we're going to take this from a skin approach and say, hey, I'm going to look at skin sarcoidosis versus do the exact same thing that I'm doing today, what we would have done was we would have used a technique called microarray analysis, or the next generation was RNA next generation sequencing. Those are all fancy words of saying, hey, we're really going to monitor what genes are up and down in this population of cells. But that's the key word. It's the population of cells. And so we would be mixing everything together, all the skin cells, all the immune cells, all the fat cells, and we might be able to detect a signal, but I couldn't tell you where that signal came from, number one. And then two, I couldn't tell you if that signal could be diluted by all these other cell populations that don't express that signal. So I'm not sure you would get, I'm not sure you would, you don't hear about all these other potential triggers for the disease. And so that was done in sarcoidosis, and that's how we got towards TNF signaling. That's how we got towards interferon gamma signaling. Those are really the major cytokines that play a role in granuloma formation. Within the last 5 years, the technology to look at the same question, but now the technology allows us to say, hey, I can actually look at individual cell types within the sample and ask the same question: what are the genes that are turned on and off? And so now that allows us to do two things. One, it allows us to characterize exactly what is, like, I think of it as a gumball machine, and that there are all these different colors of gumballs that represent different types of cells. And in the past, all I could do was compare one gumball machine to another gumball machine as a whole.
Okay.
But now I can ask, oh, well, how many purple cells are really in this gumball machine compared to that gumball machine? How many yellow cells are there? And that tells us that allowed us to figure out, oh my gosh, we found this type 1 ILC. So that was how we figure out, like, we have more yellow ILC1 cells in our sarcoidosis granuloma compared to another granuloma. And so that gave us the first signal. And then the second signal that we can say is, okay, even within these yellow gumballs, tell me what is so unique about them in terms of how their genes are turned on and off. And that allows us to figure out, oh my gosh, we found a receptor that might home these cells into the sarcoidosis granulomas. So the technology that really made a difference was single cell RNA sequencing technology. That's what I'm trying to describe. And then the next forefront is, so the next forefront, and which is what we're doing now already, is, okay, so I've talked about it as a gumball machine,
Right.
but that information doesn't tell me exactly where in the tissue is my yellow cell, is my ILC1 in comparison to the other immune cell types. And so there's a new technology called spatial transcriptomics, probably within the last 2 years or so, where basically we can ask that same question, but maintain spatial information. So now I can say, hey, are these ILC1 cells really within the granuloma center, or are they at the periphery of the granuloma, or are they adjacent to the granuloma and they're controlling how the granuloma is formed or spatially positioned? And so I think that's data set, it's still ongoing. That's people are really starting to use it more frequently now in the last several years, and as well as we are. And I think that's going to be the game, another game changer for sarcoidosis and sarcoidosis research.
That's amazing. So they might actually be like a gatekeeper. Like they're the ones that decide what gets in and gets out of that granuloma. And if they decide, if they're activated for some reason, they got to recruit all these other cells to come in and form this big granuloma.
Yeah, we think you're absolutely right. And so that's where we're missing, right? So I can tell you with the single cell technologies that we have, oh, I can tell you exactly what cell types are there. But the spatial technologies gives us a map, and to say, oh my gosh, you know, the granulomas consists of macrophages and T cells, but right next to this granuloma is this other clump of cells that seems to be really important. If I get rid of this other clump of cells, does the granuloma magically dissolve like a gate? And so these cells are really the gatekeeper cells. Give me another 2-3 years, I might have another story for you there.
Okay. That's amazing. It's so I mean, this sounds like the most promising research that has been discussed on this podcast. And, you know, and I've talked to a lot of researchers and everybody's coming at it from a different angle. But this really, it sounds like you are just so close, so close.
Well, those are very kind and very heady words. We think we're doing something right. We like that we're taking advantage of primary disease tissue and starting from that standpoint.
Right.
And then the second approach that we're really trying to use is we really prioritize having large effect sizes. Like, I want to know what are the big, I think there's going to be major signals and minor signals that are involved in any biological process. And so we're really trying to say, look, we want to focus on human samples. We want to focus on large effect sizes within the human samples. And then from that type of data, can we test hypotheses that we generate from this data in other systems to help prove what we think is going on? And so that's the approach we're using. So we think we think that there's an important role for animal research. We think there's an important role for in vitro cell culture-based systems, and those are all important experiments. We're just trying to start our questions from a human basis rather than vice versa, like starting from the animal model and then asking whether that happens in humans or not.
Wow. It's comforting as a patient to know that this level of research for sarcoidosis is happening, because we haven't had a new therapy for sarcoidosis in what, 70 years, something like that. I don't know. It's an oft-quoted statistic, and it's kind of gotten lost on me. So is there any way of knowing why it shows up on the skin versus in the nervous system, in the brain? Is like, okay, so you've got this, you know, ILC1 that's recruiting stuff, but why doesn't it always then just happen on the skin? Why does it, how does it decide, no, you know what, we need to go to the heart today?
That is a amazingly great and insightful question. And that's exactly what our next, so I just wrote a grant like maybe a week ago, and that's
Okay.
that's the exact question I'm asking. And through help through the Foundation for Sarcoidosis Research, through NIH funding, for our own sarcoidosis work, we're now getting tissue from other sarcoidosis affected organs. And so we started with human skin. Human lung is really hard to get, but we have like 1 or 2 samples. And so now we have just applied to get tissue, sarcoidosis affected tissue from other organs and from brain, from heart, more samples from lung. And we want to use the same approach that we're using in skin to answer that exact question. Can we figure out how sarcoidosis directs itself to specific organs? Is this a systemic process that it just goes awry and we're unlucky whether it hits a lung or a brain or a heart or skin? Or are there specific changes within the sarcoidosis cells, immune cells, that really target a specific organ? We don't know. All we're finding right now are the major signals that are building the granulomas. I hope to find the organ-specific signals as well as we move forward.
That would be so amazing. So we look at the menu of drugs that are being used to treat sarcoidosis right now. And the front line is prednisone, although methotrexate apparently is coming on is potentially equally effective. There's been a lot of discussion of that lately. And then you've got all the biologics out there that work. None of them are directly have been developed directly for sarcoidosis, but they seem to work, and I think they all use a different mechanism for working. Is there anything here that you've figured out that says, well, logically this is why methotrexate works, or logically this is why prednisone works?
So certainly, I would say that prednisone. So I think prednisone is a general immune suppressor, right? And so that's a very broad weapon to just knock down your whole immune system. And that's why it's so effective in treating sarcoidosis. Methotrexate is a steroid-sparing immune suppressant as well. So similar class. So these are great drugs and that are well established. The downside about them are the downside side effects that we know happen when you're on prednisone for longer than 30 days or even 2 weeks to 30 days. You know that you'll have a weight gain. You know that you'll have a higher chance of osteoporosis, a higher chance of diabetes. And these are inevitable side effects. These aren't side effects that may or may not happen. They're just inevitable because the drug hits so many different signals. And so the next generation is that we can make more targeted therapeutics that give that same effect of keeping your disease in complete remission or treating your disease completely, but also having much less severe side effects. And so from our own work, I would say that, look, we're far from it. We need to do clinical trials. We need to formally prove this. I wouldn't want anyone to misinterpret what I say next, but that target that we found, CXCR4, that homes the ILCs to the sarcoidosis granulomas. Well, guess what? There is an FDA-approved drug already being used in patients for a different indication called plerixafor. And this plerixafor is an antibody that blocks CXCR4. And so theoretically, if we give sarcoidosis patients plerixafor, we may be able to help treat their disease in a more specific and targeted way. So we're trying, and so the benefit here is that what we're doing here is not trying to discover a new drug. And then when you discover a new drug, that's at least a 10-year clock that before the drug ever gets to a patient. Here we're saying, hey, we want to repurpose this already existing drug to try it in a new indication here for skin sarcoidosis, in particular for us. And so we're running that clinical trial now. So Dr. Rosenbach really is running this clinical trial here at the University of Pennsylvania, trying to use this drug to test whether it may affect skin sarcoidosis granulomas.
Got it. So you are a researcher, but you also see patients as well, right? So, and do you see primarily or only skin cases? You're a dermatologist,
I'm a
right?
dermatologist, right? I see patients. So I have what I think is the best job in the world. I see patients half a day a week. Half a day a week. I see general dermatology patients. I help train the next generation of dermatology residents in our program, and then the 4.5 days a week, I'm in the lab trying to do these, trying to improve precision care or personalized medicine for dermatology, including sarcoidosis.
Got it. Got it. Got it. Got it. So you guys are looking at all these. Is there another aspect of this that we haven't talked about that that you want to look at next, or that you're looking at on the side when you're not exactly doing this, does this take 100% of your time?
Uh, um, that's a real, so I would say that the first half of the project of ILC1 and really following up on whether this is a true biomarker, a diagnostic biomarker, that's taking up a lot of our time because we're trying to formalize it into a blood test, right? And so right now, sarcoidosis is a disease, a diagnosis of exclusion. You have to rule out all these other things before we can even give you a diagnosis of sarcoidosis, and that delays diagnosis, delays initiation of treatment by months. And so we're hopeful that we can use this ILC1 immune cell that we found as a clinical test to say, hey, now I can actually help you by just drawing your blood or by biopsying your skin. I can say whether you have a higher chance of sarcoidosis or not. And so that's taking a lot of work. That's a formal clinical trial. That's recruiting over 200 patients. And so that's the bulk of our work. The second half is what I just talked to you earlier about using CXCR, trying to repurpose this existing drug for treatment. And then the third part of our work right now is on this blood clotting link and that protein that we found, osteopontin and thrombin inhibitors, because we think that ILC1 is a diagnostic biomarker and is a important way to help organize your granulomas. We think this thrombin osteopontin system is really a major engine of how granulomas form for all diseases. And so if we can target those things, that might be an alternative way to treat the disease. So we think we're approaching granulomas and sarcoidosis from at least 3 different directions right now. I believe that focus is important in order to really make things happen, and so rather than spreading ourselves too thin, we're trying to see these 3 things through first, and then we'll evaluate other possibilities.
So this ILC1 as a biomarker, and the fact that there could be a blood test that could diagnose Sarc patients, that would be so much less invasive. I mean, it could be something that could be ruled out very early in the diagnostic process, right? Just let the computer look at the blood and spit out all the possible things it could be, and then a physician could look at it and say, "Oh, I think you might have sarcoidosis." Is that a reality in the next 5, 10 years, or are we dreaming?
Um, that's a great question. So the conservative side, okay, so that's a great question. I hope it is a reality in the next 5 to 10 years.
Okay.
And here are the barriers. The first is that in order to detect ILC1 right now, that specific immune cell type in the blood or in the tissue, you need a lot of specialized equipment that aren't available in all hospitals around the country. They're available at the major academic centers, they're available in research labs, but they're not everywhere around the country. And so what we need to do is, first we need to validate ILC1. The second thing we need to do is try to convert that sort of complicated test into a test that can be given at any clinic, given at any medical center, either through at any clinic or medical center. And so we're actively looking at ways to do that as well. And so
Yeah.
if that works out, 5 to 10 years would be reasonable, but there's a lot of work there still.
Yeah, so now when I get a blood test, I go down to the lab and a technician with community college training takes my blood and fills out the vials and writes my name on there and sends it off uh, to whatever lab they send it to. But that lab likely is a long way from being able to see if there's an ILC1 biomarker in it.
Yeah, right now that lab is farther away than I would like, but we're going to try to actively try to shorten that distance.
That's fantastic. Um, and then let's just get back to the, using skin. Is there anything else about our skin other than ease of access that you know about skin that the rest of us don't know that makes it the perfect place to study this?
Um, it so and this is not my own work. This is work for of our field, I would say,
Okay,
be that the understand so the idea of using skin as an entryway to study systemic diseases, like
right.
systemic diseases, by just we'll learn a lot by just studying the skin, will can apply to systemic diseases. I think that has been a theme that has been um, continuing to grow over the last decade or so. And
Okay,
and the best example of this would be melanoma research as a model for tumors and oncology. We've had a lot of drugs being developed for melanoma, obviously, which is a skin cancer, and the reason why is um, many of the principles by studying melanoma can be applied to colon cancer and lung cancer, and um, because because it is really difficult still to get heart biopsies. It's really difficult, well, for obvious reasons, and it's really difficult to get lung biopsies because it's just even more invasive of a procedure. Meanwhile, you come to a dermatologist's office. If anyone's ever gone to a dermatologist's office and we see a lesion on your skin that we're concerned for malignancy, it's a 5-minute procedure for us to take it off. And so that barrier of entry of studying the actual disease rather than inferring it from a blood sample or inferring it from um, liquid that you collect from the lung. That's there's a I think we think that there's a big difference there, or we and others, not just myself, think that there's a big difference between studying the primary process and or and having to infer and having to take one step further away. I'm not saying that that's not the right way. I'm not saying that those ways are not appropriate or not good. They are. We just think that by studying the actual tissue itself, we're getting an additional advantage and make perhaps helping us figure out molecular drivers more directly.
So a lot of the patients who have come on the podcast are big time either endurance athletes or workoutaholics, people who led extremely active lives. And I don't know if they have found me because that's kind of who I am and I've talked about it a lot, but it seems like it's more than a coincidence to me with my very small sample and no medical training. But it's like everybody comes, oh yeah, I was a marathon runner. Oh yeah, I was doing this, 1 day through hike across the Grand Canyon and everything blew up. Or, I'm a trail runner. I've had so many of those types of people come on the podcast, and I correlation, causation, whatever. I don't know. There's nothing about your research that would tie back to any of that, is there?
I'm trying to think it through. I love the idea. I have not heard this correlation so strongly.
Yeah, yeah.
And so and now I'm even more intrigued, and I'm going to start reading about this when we finish this
Okay.
podcast.
All
And
right.
I think I can think of so the so in addition, so I think what makes our work unique, Dr. Rosenbach and my work unique, is that um, So we're really good at the genomics. We're really good at the human samples, and that's great. But that only leads you so far, because that really leads you to correlation, what you just said. I can
Mm-hmm.
correlate this. In order to really prove the causation, you have to test your hypothesis in another system. And so we've built mouse models, or animal models or mouse models. We've built cell culture systems to test our hypotheses and to prove causation. And so perhaps I need to make sure I get some animals that are in top physical fitness, and
There
then
you
do
go. Right.
our animal model granuloma formation, and test if there's a difference. It sounds really intriguing to me, and something we could certainly test.
Marathon mice.
We will include you in our author list.
Okay. Is there anything else you want to add, Dr. Leung?
I mean, I think my parting, if I had to say one parting thing, would be, I think science is moving us closer to truly understanding sarcoidosis. I think we're uncovering not only just what happens in the disease, but why it happens. I think that's really important. I think there's hope for patients on this horizon. I really think that in the next 5 to 10 years, we're going to be turning scientific discoveries into more precise and effective treatments than we have now. And then the last thing I'm going to say, I've been hesitant to say this, but I think I have to say this. We can only do this type of work in science if the world continues to support it. Right now, research funding is really under tremendous pressure. I think part of the problem is, as scientists, we haven't done a good enough job showcasing how our work has really, truly impacted patients' lives. And so I'm very grateful to you, John, this podcast, and the Foundation for Sarcoidosis Research for helping us share this story and for giving science a voice.
Yeah. Can we look for some sort of a poster or an article soon from you guys?
We certainly, our work on ILC1 was published in the Journal of Clinical Investigation.
Okay.
You can find all this information out on my own lab website, which is thomasleunglab.org. And that's probably it.
No, that's great. Well, I'll link to it. That's why I asked, because I'll link
Yeah.
to it, and we want to be part of the solution here. So if we can continue to shine a light on your research or do whatever needs to be done to point dollars in your direction, which follows, people having a fundamental knowledge of what you're doing and the success of what you're doing, then we want to be able to try and do that for sure.
Thank you so much for the support. Thanks for this opportunity. Really appreciate it.
Yeah. Thank you.
I feel like a zombie, just feeding and stumbling.
So thanks to Dr. Thomad Leung for joining me here on the FSR Sarc Fighter podcast. Wouldn't it be great if there was a biomarker in our blood that we could just get a blood test and someone could say, oh yeah, it's sarcoidosis? That would be amazing. And then his gumball analogy, you got two gumball machines and some of them are yellow and some of them are blue, and all of a sudden there's a whole bunch more yellow gumballs, and why is that? And I thought he did a really, really good job of making all of this user-friendly, talking about this homing signal that's attracting all of these immune cells that you don't want attracted. And, I still ask myself, what was it about my spinal cord that sent out a homing signal, and then all this stuff just started accumulating, for me and my spinal cord, for most people in their lungs, for a lot of people, cardiac sarcoidosis. But it seems like we are well on our way to getting to the next level of understanding. So thanks to Dr. Thomad Leung for joining me, and more importantly, for all of his research. This is a good time to contact me if you'd like to share your story on the podcast. The best way is to email me at carlinagency@gmail.com. The official Sarc Fighter song is Zombie by Mark Steyer and his band, the White Hot Lizards. Mark's a fellow Sarc Fighter. I release this podcast every other Monday. As I'm speaking today, my trusty dogs Dougal and Shandy are not in my office, although they were, and they just got up and left a little while ago. But together they make my life so much better. I love my dogs. The backstory to the founding of the Foundation for Sarcoidosis Research is episode 11 with Andrea and Redding Wilson. Please follow Sarc Fighter on social media, Facebook, Instagram, Peloton. Check out my cycling blog, Carlin the Cyclist. There's a section there called Cycling with Sarcoidosis, and there's also lots of writings about my various travels with the bicycle. You might like it even if you don't read the articles associated with about sarcoidosis. If you're new here and you're trying to just figure out what sarcoidosis is, listen to episode 2 with Dr. Simon Hart. My story is episode 1, and there is a link to a television story about my case that's also in the show notes with the description of this podcast. Please send me an email. It's in the show notes. And until next time, keep fighting.
Learn to suffer, you feel pain someday. Learn endurance, your strength will fade away. Dead man walking, trying to keep up the pace. Dead man walking,